Now, there are 4 models that we use. Yes, there's 5 things up there because the last is us. But we don't use humans as a model organism because the model organism, what it's modeling is us. When we use vertebrates for study and development, again, it's about us. It's about learning how humans develop without actually having to do experiment and genetic studies on humans. A few people have done that in history, and they're not very nice people. Xenopus, fish. So Xenopus is the frog. Fish is the zebrafish, which the species is Rario. Chick we call Gallus is the species. Gallus is the domesticated chick or chicken. And then mouse, Mus musculus, is the genus species for that. And then Homo sapiens are humans. So these are the 4 vertebrate model organisms that I'm going to talk about: Xenopus, fish, chicken, mice. So the first thing we're going to talk about is the advantages and the disadvantages, just like we talked about before with the invertebrate models, on why we use them for various purposes, and then, of course, what we learn from each one of them. So that's really what we're about. We're answering the human questions of development. So let's start with the Xenopus. Here are the advantages. The cells are so large you can actually see a frog embryo with the naked eye. Now, you can see a tiny little dot, but most cells you can't even see with the naked eye. So because the cells are large, we can see a lot of the micromeres and the smaller cells that develop as a result of that. That's one advantage, is the cells are so large we can look and watch the development of these in a great amount of detail. Frogs, in their earliest stages when they become tadpoles, have very rapid development. And obviously, it slows down after that, and then they'll go through a metamorphosis into the adult stage. But in the initial stages, when they first develop into the tadpole, it's very rapid. So the more rapid turnover of an organism, the more we can study its overall development, which is a great advantage. We use frogs primarily for transplantation experiments. So the moving tissues from one area to another to answer the questions of specification and commitment of these cells. This is where we've learned quite a bit about that. Now, we can use other organisms for this as well, but frogs in particular are more advantageous for these transplantation experiments. Now, unlike Drosophila, which are great for genetic studies, Xenopus are not very great for genetic studies because, like us, they have multiple chromosomes. And it gets very difficult to show one-to-one relationships between mutagenesis of a particular gene and the resulting developmental defects that come from that. Drosophila and some of these invertebrate model organisms are great for genetic studies because of that one-to-one relationship between a mutation in the gene and the development of certain regions. But frogs start to get more complex, like us, in the multiple chromosomes, as well as the way in which they regulate their genetics, as we know, as far as gene splicing and things of that sort. Another disadvantage is you can't go through many generations with the frogs because they go through a long growth period before they're fertile again. So unlike Drosophila and some of these other organisms where we can keep breeding them generation after generation and look at the long-term effects of mutagenesis and other genetic studies, frogs aren't great for that. So that's really the advantages and disadvantages of using the Xenopus. Most of what we're going to look at with the Xenopus are commitment, looking at the cells in their specification process, when they become committed to the various germ fates and the germ lines and then the cells in that regard. So that's what they're really good for. Fish models, or the zebrafish, is mostly used for ours. Lots and lots of embryos. They breed all year long. They're easy to go from one generation to the next. Zebrafish can be bred in the lab, and so they're good for generation after generation. Similar to the sea urchins, their embryos are transparent, so we can see a lot of the gastrulation processes that are occurring in the fish model. I showed you that video before, and I'll show it to you again, where it actually shows the time lapse of the embryo developing, and you can see almost everything going on during those stages. So they're very great for looking at gastrulation. They develop outside of the mother, and the reason why I bring that up is because you'll see here in just a second regarding the mammals. That's one of the biggest disadvantages of mammals is it develops inside, and you can't really watch its development as you can with the fish models, with the Xenopus models. And then one of the big advantages, one of the things that we struggle with at times when we're doing genetic studies is integrating genomics into the cells. One of the nice things about this fish model is the cells are very permeable to some of the lipofectamines and other ways of introducing genetics, recombinant genetics into the cells. So they're great for fate maps, being able to tag them with GFP so we can look at where genes are being expressed. These are, so the fish model's been used substantially because of that, the Xenopus. Chickens. This is what I primarily deal with. I'm the chicken man. Disregard Toy Story. But we actually are doing chicken embryology for the cell biology lab, where we're going through the various stages, and we incubate them, and we're able to see them at the various stages throughout their whole development. Easy to raise outside. You can just constantly, we're getting eggs all semester long, and so we're able to breed them in the incubator. One of the great things about the chick model, or the chicken, is that we can time and know exactly what stage we want to look at just based upon temperature. All I have to do is put it at the right temperature and a certain number of hours, and boom, we know exactly where we want it to be when at development. So they're very predictable. Sometimes, because of fluctuations in certain parameters in some of these models, you can't always time their development. There might be differences in some of the stages, but chickens are very easy to be like, hey, I want them at when their heart starts forming, like we did yesterday, and I know exactly how long to incubate them for that amount of time. One of the fascinating things about the chicken models is they're more similar in their earliest stages of gastrulation to humans than mice are. So especially in the earliest stages of movements in the formation of the 3 germ layers, chickens are almost identical in the formation of the 3 germ layers and how that's patterned as humans are, which is a huge advantage for us to be able to learn more about ourselves and using the chicken model. Another thing that's great about chickens is because they're in ovo, they're in the egg, we can not only do experimental transplantation, but we can also do genetic manipulation. They're very easy to manipulate genetically, and I did a number of genetic manipulation studies for my PhD, as well as some experimental manipulation. So they're great for both of these. You can manipulate them, cover them back up, and let them incubate longer. So you can just stop, do some things with them, let them keep growing, and then go look at them later on. They're absolutely wonderful which is why I chose them, for a model. I did have a choice in my PhD to use a mouse as a model, but I said screw that, because it takes months for you to get enough organisms to even do an experiment. And I had a guy on my committee. It took him 7 years to do his PhD. It took me 3.5. So what model you choose to study, I was able to do experiments with the chickens after 2 days, where it would be the equivalent of after 2 months with mice to get the same thing. So I chose wisely and able to finish my PhD quickly. But he took 7 years in studying development using the mouse model. Now, mouse model does have advantages over all of the others. And a lot of the advantages are in terms of genetics and later development, they're the most similar to humans. So obviously we need to use them as a model because their genetics and their development past a certain point are so similar to us that how can we not? They're very easy to maintain within a housing environment, very easy to breed from one generation to the next. You can just keep these lines going of these mouse models back in most places except for here. we've always had a mouse facility where we've been able to breed them and do a number of genetic studies. Disadvantage, you don't get a lot, and it takes forever to get enough to work with to do some of these studies. You also don't get to manipulate them as easily as you do these other models because they're developing inside. Pretty much once you take them out, you're done. You can't put them back in. And a lot of the embryos are so small, it's not advantageous in terms of watching this. So there's several factors against the mouse model. But the trade-off, because of their similarity to our development in terms of genetics and later stage development, you can't ignore them. You have to use them as a developmental model to be able to understand us better. So these are all of the advantages and disadvantages of these models. Now let's get on to fertilization. All right, let's talk about fertilization. Now, we've already talked about some of these things, but now we're going to get into more detail regarding how fertilization in these model organisms initiates gastrulation, cleavage and gastrulation in these embryos, as well as how even some of the earliest stages of fertilization, or the fertilization itself, leads to axis specification. Because in some of these organisms, where the sperm enters into predetermines the axis that is going to develop, from the ventral to the dorsal to the anterior to the posterior. So even at the earliest stages of fertilization, we're going to talk a little bit about axis specification in each one of these embryos. So I'm going to go through each model organism, and we're going to talk about how fertilization and some of the key events of fertilization. Let's start with Xenopus. The way that the frog embryo is set up, or the oocyte is set up, the sperm can only enter in through the animal pole. It cannot penetrate the vegetal pole, or where most of the high concentration of yolk is at. So it can enter into anywhere along the animal pole, any part in the animal hemisphere. Now, one of the key things that, if you use Xenopus in talking about fertilization, I don't want you to just describe this, but cortical rotation. This is a huge thing in terms of axis specification, as well as in the initial patterning of the embryo. Let's talk a little bit about cortical rotation. What happens is, in the animal pole, the sperm will enter into any part of it. Now, where the sperm enters into predetermines the ventral region of the Xenopus. So 180 degrees, or on the opposite side of where the sperm enters, that's the most dorsal region of the Xenopus. So even axis specification is predetermined by where the sperm enters into. We'll get into the dynamics as we go through this lecture on how that occurs. Well, one of the first things that occurs is what we call cortical rotation. Now, at the most vegetal pole of the Xenopus oocyte, one of the maternal factors that is sequestered there is disheveled. Do you remember what signal transduction pathway disheveled plays a main role in? Wnt signal transduction pathway. So disheveled is found here, but what happens is the outer region of the oocyte shifts exactly 30 degrees. I mean, that's amazing in and of itself, the exactness of it. But always 30 degrees. It'll shift the outer layer, which then pushes this disheveled, which is at the most vegetal pole, to one side. Now, that region right there is going to be a key region later on in the gastrulation process of Xenopus. We haven't talked about the organizer yet, but just to give you a preemptive, that's where the organizer is going to form, is right there. So as far as fertilization goes, the sperm can enter anywhere into the animal pole, but where it enters into predetermines the the ventral region of the Xenopus. And the other side is going to be the dorsal region of the Xenopus. I'll get into the dynamics later on of disheveled when we get into the organizer, but that is already, as we've mentioned, this is where you get asymmetric distribution of various maternal factors that are going to influence after cleavage occurs and a lot of the gastrulation movements. So let's talk about fish fertilization, Xenopus. We've talked about the calcium waves that usually come about because of the, in fertilization, these calcium waves will help initialize some of the maternal components and allow the genes to be translated into protein and then start the ball rolling. So these calcium waves are part of not only to prevent polyspermy, but they're also meant to initiate various processes within the cytoplasm. One of the processes that's important for fish is that the cytoskeleton will actually squeeze part of the non-yolky area and create this little bubble, which we call blastodisc, on the animal pole. So this apical blastodisc is where cleavage is going to occur. Chick fertilization. So ultimately, chickens can lay eggs and not have them fertilized. Okay? They don't have to be fertilized for them to lay the eggs. When the chicken lays the egg, as it is traveling down the oviduct, the albumin becomes surrounded by a calcified shell, and then eventually the egg is laid. Well, fertilization has to occur internally before the shell is secreted to cover the egg. So you have the albumin, you have the yolk, and then you have the oocyte. Well, it needs to be fertilized, and then the shell gets secreted around that. In fact, as the egg starts traveling down the oviduct, forces of gravity will play a role in the patterning, and the anterior-posterior axis patterning of the embryo itself. So the mere gravity that will cause certain maternal components to be sequestered to one side are critical in the anterior-posterior formation. The fertilization has to occur internally, similar to us, obviously, before the egg is laid. It doesn't occur after the fact. I'm surprised that not many people know this, but there it is. Okay, so mammalian fertilization. There's some fascinating things about this includes mice and rats and humans. It occurs in the oviduct. Now, one of the fascinating things about mammalian fertilization is when the female ovulates the oocyte, meiosis is halted at about metaphase 2, and it won't even finish until fertilization. So once fertilization occurs, then the last stages, anaphase 2 and telophase 2, and then the removal of the polar body, which is the second nuclei of the oocyte, does not occur until fertilization occurs. In fact, it won't go past metaphase 2 unless fertilization occurs. So this is something different that you would find in mammals rather than in other organisms. And then cleavage begins as it's traveling down the oviduct. In fact, it has to be ready to be implanted into the uterus by the time it reaches the uterus. And so there's a lot of cleavage and gastrulation going on as it's traveling down the oviduct before it's able to be implanted into the uterus. So that's fertilization. This is one of the easiest of the four. Okay? So I just explained some of the initial mechanisms. Now, I'll explain more about fertilization later on and as it relates to these other topics as we go through here. But here you can see after ovulation, the oocyte still is right in the middle of metaphase 2, and then it will be fertilized. It will then finish meiosis 2, eject one of the nuclei, which we call a polar body, and retain the last one, and then those nuclei will fuse, and then it will start going through the processes of cleavage and gastrulation, and then eventually implantation into the uterus. Cleavage. Now, we've talked quite a bit about cleavage, so this will kind of be a recap of what we've already discussed, but there will also be some new things that we're talking about as well. You already know that the yolk in various organisms inhibit where cleavage occurs. In regards for the model organisms that we use, it affects 3 out of the 4. In mammals, there really isn't any yolk. So the cleavage is holoblastic and completely divides all of the cells evenly. However, for frog, fish, and chickens, there are some fundamental differences that I need to reiterate and go over. In the Xenopus, as well as in the zebrafish, the animal pole is the area that has the low yolk, and the vegetal pole has the area where there's the high yolk concentration. So let's look at the Xenopus first. Its mode of cleavage is displaced radial holoblastic cleavage. Now, holoblastic, remember, means what? Entire cytoplasm. Everything undergoes cleavage. There isn't a part of the oocyte that's left out in the cleavage process. Displaced has to do with the fact that because of the different concentrations of yolk from the animal pole to the vegetal pole, then the blastomeres, or the cells that result from cleavage, are going to be smaller in the top and larger in the bottom. You're not going to get even division amongst the cells. The ones in the vegetal pole are going to be much larger than the ones in the animal pole because of the different concentrations of yolk, which is going to impede cleavage in the most vegetal regions. In fact, the first few cleavages don't even complete before the next ones start as they're going down. In fact, it creates these stress folds in the animal pole. So cleavage occurs kind of from the animal pole down to the vegetal pole in the first few cell divisions, and you can see how the first one's not even done and the second one begins. Now, you do have mitotic divisions going on when you have multiple nuclei in these particular regions where the cells are going to be partitioned, but these stress folds are going to happen all the way down here because as it's trying to pull the membrane in and completely close off these, you can see that it's easy here in the animal pole. It's much harder in the vegetal pole. That'll create smaller blastomeres in the animal pole and much larger blastomeres in the vegetal pole due to that differences in the yolk concentration. So you can see that here as well. This region right here, this is what we call the gray crescent. Now, the reason why this is called the gray crescent is a frog embryo is really dark on the animal pole and much lighter in the vegetal pole. Well, what's one of the first things that happens after fertilization in the animal pole? Cortical rotation. So as the cortical rotation occurs, it actually creates this area which we call the gray crescent, which is a little bit lighter in color. It's not going to be as dark as the animal pole because of that cortical rotation. This is going to be a key area because in this region right here is where most of the initial stages of gastrulation occur in the Xenopus. This is a key area for organization of the frog embryo. But one of the key things you have to understand about the blastoceles, you'll see it in pretty much all of these organisms, in the not only the gastrulation processes where the cells are moving, but also in the specification process. If this blastocele is not there, the three germ layers will not form properly. There needs to be a separation, especially from these most animal pole cells, or the most these cells right here, from these cells, because there are paracrine factors and various interactions and morphogen gradients that are being established here that if you don't have this blastocele, this will not form into ectoderm and neural tissue. All right, zebrafish. It's discoidal, just like Xenopus, but it's meroblastic, which means what? Part of it. Meroblastic is just part of the embryo. So as we've talked about in the first parts of fertilization, the actin filaments will squeeze the animal pole, forming a blastodisc, and this is where cleavage is going to occur. It's going to start forming this mound of cells on the animal poles above the yolk. There will be nuclei that will be just beneath where cleavage is occurring, and we call this the yolk syncytial layer. Remember syncytial specification, where in just beneath this layer, in fact, the nuclei will eventually surround and still become this yolk syncytial layer. There will be nuclei just underneath that are important in the development process. So here, this is the blastodisc. It undergoes discoidal meroblastic cleavage. In fact, in these first stages of cleavage, the underlying area, the cytoplasm here, is still in contact with the yolk. There are key factors that will come from gene transcription in the nuclei that are in this yolk syncytial layer. In fact, let me see if I can, oh, here it is. So here, eventually the cytoplasm of these cells will undergo cleavage and they will close off. But initially, very similar to the Drosophila, where you have some morphogen gradient, this cytoplasm needs to remain open to allow some of these paracrine factors to help in the specification of these overlying cells. Eventually, all of these cells will close off, and then it'll continue to undergo cleavage until you form this huge mass of cells, where the inner cell mass, very similar to you and I, the inner cell mass, that will form the embryo. And the outer region, this will form a shield that is necessary for the envelopment of the yolk during the gastrulation process. But the main part here is this blastodisc, and this is where cleavage is occurring. It is exposed to the yolk syncytial layer during the initial stages. We'll talk more about that when we get into axis specification. Eventually it closes off because it's received all of the paracrine signaling it needs from that yolk syncytial layer, and then the cells will start undergoing gastrulation. So those are the initial stages of cleavage. Here's another picture just showing you. Here are the yolk syncytial nuclei here. Here's the enveloping layer, the overall blastoderm, but it's only the deep cells, or the inner cell mass, or these deep cells are what become the embryo. The enveloping layer will form a shield around the yolk and play a role in development, but it won't actually become the embryo itself. Similar to the trophoblast in our development. Now, chickens, they also have discoidal meroblastic cleavage, identical to zebrafish in terms of how we call it discoidal meroblastic, but there are some fundamental differences. One of the things is that it doesn't form a bubble on top of the yolk. It just starts undergoing cleavage right at the top layer of the yolk. So it does form a blastodisc, but not like the Xenopus where it squeezes it and forms this blastodisc. You're only going to start seeing, if you look top down, you can see that in the topmost area of the yolk, you start getting cleavage occurring. And these cells will essentially just form one layer on top of the yolk. So it's meroblastic because it's not going to encompass all of the yolk, and discoidal because you're getting only a portion of these, and these cells are going to start forming the initial germ layers. Now, the blastocele forms just beneath here. In fact, these cells will cause what we call a subgerminal cavity to form. These top cells will actually form a subgerminal cavity and then form another layer of cells beneath it, which will form the blastocele, which plays, again, a role in gastrulation in a lot of these movements going on. So what that does is it creates an area that is much lighter. When you look at it, it's much more clear. We call it the area pellucida. And the reason why it's much clearer is because water is between the yolk area and these cells that are forming, and therefore it's a little clearer to look through. The area just outside of that, where cells are also forming, is called the area opaca. And now if you think of opaque, it's kind of foggy or whatnot. That's because this is flush with the yolk, and so there is no separation. So that's why this area is a little bit more clear, is because there is some fluid just beneath this top layer that gives it more of a translucent look. We call that the area pellucida. That's where the embryo, the main embryo, forms. There are going to be cells and other things that form out here in the area opaca, but those are mostly extra embryonic. They're not going to form the main embryo itself. So cleavage occurs just in a very restricted area in the area opaca, though there is other things going on in this other area called the area, in the area pellucida is where the embryo forms, and then you have the area opaca where it's flush with the yolk. There's no subgerminal cavity. There's no blastocele that's forming just underneath these cells. So this will eventually form a second layer. The first layer of these cells is what we call the epiblast. So the epiblast that's what forms the embryo. That will actually form all three germ layers, is these cells. The hypoblast, which is a group of cells that forms just beneath the epiblast, actually do not become the embryo, but they're necessary in the patterning of the embryo from the epiblast. So the hypoblast plays a key role in axis specification and in specification of certain cell types during the development, but eventually the hypoblast gets displaced by cells from the epiblast. So as far as understanding cleavage goes, the epiblast, which is that first top layer that forms on top of the yolk, is the embryo proper, or the actual embryo itself. It's going to form all 3 germ layers. So the biggest mistake people make is thinking that the hypoblast forms the endoderm, and that is not the case. The hypoblast does not form the endoderm. If you look in the top side, you'll actually be able to see here in the area pellucida, these cells start, we'll talk about gastrulation here in a little bit, but the cells start migrating more anteriorly, and then it starts forming the 3 body axes on this region. Eventually, you get this region called the primitive groove that forms the anterior to posterior axis of the embryo. And we'll get into some of these movements later when we talk about gastrulation. Mammalian cleavage. Holoblastic, isolithal. Iso means the same. So all of the blastomeres that are going to come from the cleavage processes are going to be the same size. There really is no animal and vegetal pole. There's no differences in the yolk. There's no yolk, really. Now, it's rotational because it doesn't divide the same way each time. In fact, it changes the plane in which cleavage occurs. So it's not similar where you always have it along the same axis. What you'll find is that in amphibians, you get perpendicular cleavage in this regard, but over here, or I'm sorry, you get them all along this, totally like having a brain fart right now. Anyway, you can see the different cleavage planes here versus here, where this starts becoming perpendicular to the initial cleavage plane. So this is what it means by rotational, is the actual cleavage plane rotates and changes in these initial stages. Now, holoblastic because it encompasses the entire oocyte, and you get cleavage pretty much right away. Now, one of the interesting things about mammals that doesn't occur in other organisms is after about 8 cells form, you get what we call compaction. What compaction is unlike other organisms where you can actually see the individual blastomeres forming, after a certain point, the cells will increase these cadherins, or the cell adhesion molecules, and the cells will seem like one large lump. They'll actually become so tight that you can't tell the difference from one cell to another. So here's about the 8-cell embryo, and this is where compaction occurs. See how they start fusing together almost? Now, they don't actually fuse together, but they start forming such tight junctions, but you can't really see some of the divisions between the cells. The main reason for this is because they start forming gap junctions for cell-to-cell communication. They need some cell-to-cell communication for these earliest stages in this development process. So that's really what compaction is all about, is that the cells need to have a greater contact with one another. They increase the cadherins, these cell adhesion molecules, and they start forming a tighter mass. So you can see here in the mouse embryo, initially you can see a lot of the cells, and then compaction occurs, and you can't see really the different cells anymore. They're there, but you really can't see a distinction between one cell and another. Eventually, the blastocele forms, just like in other organisms. The blastocele, again, plays a major role in gastrulation and in various movements. So this region right here, we call this the inner cell mass. This is what becomes you and I. So these cells right here are what become you and I. The outer cells we call the trophoblasts. These form the extra embryonic tissues, such as the placenta and the umbilical cord. So these are things that are necessary for development, for the implantation into the uterus and things of that sort. However, these cells right here are what become you and I. When you have monozygotic twins, this mass will split at an early enough stage that the two masses then become two completely separate organisms. Now, on occasion, they may not split completely, and you'll get twins that are conjoined and things of that sort. If they do split, there are a number of ways where they can share the same amniotic sac. Sometimes they don't share the same amniotic sac. You know, there's a number of things that can happen after that inner cell mass splits. They can share one placenta. They can each have their own placenta. So there's a variety of different combinations. But the long and the short of it is monozygotic twins have the same genetics because they arose from a splitting of that inner cell mass which is just mitosis. So they have the exact same genetics initially, but there's a lot of environmental factors that change their features as well. Even monozygotic twins don't have the same fingerprint. All right, now into gastrulation. This is where we're going to spend a lot of time, as well as in axis specification. Here's where we really start getting down to the dynamics of how things form. In fact, most of the rest of the semester will be on these two. When we start talking about neural development, we'll talk about how gastrulation forms the brain and the spinal column. And we'll talk about how the muscles and the bones form through gastrulation. As we're talking about gastrulation of the brain and the spinal column, we'll also talk about the genes involved in specifying those cells to become, the trigeminal nerve and the various vestibular cochlear and all these other types of nerve bundles and fibers. So these play a role hand in hand for pretty much the rest of the semester, minus the last two lectures where we're going to talk about medicinal. And then what are some of the dynamics regarding, the genes and the signal transduction pathways. Those are going to go hand in hand in each one of these development of the organ systems that we talk about. So this is just going to give you a prelude to pretty much the rest of the semester, these two, which is why we're going to spend quite a bit of time on this. Now, the first thing we have to know about gastrulation is in vertebrate gastrulation, they pretty much have some of the same things in common. One thing that has to happen in all vertebrate gastrulation is in the initial setting up of the oocyte, you have the ectoderm, mesoderm, and endoderm. Well, the endoderm and the mesoderm have to be internalized. So some of the first movements of gastrulation in any of these organisms is the internalization of these two germ layers, the mesoderm and the endoderm. They start off initially on the outside, but they eventually have to end up on the inside. That's one of the things that we'll show in each one of these modes of gastrulation is how do they internalize cells to become the mesoderm and the endoderm, even though fate maps have shown that, for example, for this frog, the fate maps show that the ectoderm and then this will become mesoderm, these regions right here, well, mostly over here, but this region will eventually become the mesoderm, and then the endoderm will be completely enveloped by the ectoderm. So the ectoderm will form the outer layer, the mesoderm the second, and the endoderm the most internal layer. That's one of the things we'll show in each one of these gastrulation movements. How is that accomplished? Well, pretty much in all cases, you have epiboly. If you remember, epiboly is the movement of cells over another set of cells. So the ectoderm typically undergoes epiboly around the entire embryo. And that's for fish and for frogs and for mammals and for chickens and birds and all that kind of stuff. Another process, which is kind of the same thing as epiboly, but not, it's called convergent extension, where the cells not only are undergoing movements, but they're also undergoing mitotic divisions. And so what you'll see is that as the cells become internalized, you'll start to see multiple cells increasing, and so it'll look like it's expanding, which it is, because the cells are accumulating more nutrients and they're undergoing mitotic divisions as epiboly occurs. And eventually, they'll bring a lot of these cells into their various germ layers, the ectoderm on the outside, mesoderm, and then endoderm. And then finally, you'll typically get the first indications of the anterior-posterior axis forming in the earliest stages of the embryo, where it'll start forming the body axis, anterior to posterior, dorsal ventral, left, right. It's easier to see this in organisms like Xenopus and chickens and humans. It's a little bit harder for zebrafish as well as for mice. So we'll do the best we can as we go through here. But these are four kind of movements that are common in all vertebrates. But beyond that, the ways in which they get there, there's a lot of differences. So there is going to be some comparison between the model organisms and how they're similar. There's also going to be some looking at some of the fundamental differences between each of these. So let's talk about Xenopus gastrulation. Now, the gray crescent, if you remember, that's the region that after cortical rotation, that you get this tiny gray area. So right there, that gray crescent right there. This is where the first gastrulation movements occur. So right here at this gray crescent, the first thing that happens is there are some cells which we call the bottle cells. The reason why they call the bottle cells is because they're kind of oblong in one area and fatter down in the area. They look like a bottle. These bottle cells start the what we call involution process. Do you remember what involution is? It's where one set of cells will move underneath another sheet of cells. That's one of the gastrulation movements. So right here, this is where the gray crescent occurs. These cells are a group of cells, which we call the bottle cells, that will start involuting and start the initial stages of gastrulation. Now, you can see this right off the bat. This is what we call the dorsal blastopore lip. Here is what's happening right here. Here's the electron scanning micrograph showing you how these bottle cells will start involuting. This creates the dorsal blastopore lip. These cells will start moving underneath these. Now, the blastocele is right about here, underneath all of these cells. So this is going to start moving just right around the blastocele of the embryo. That's one of the reasons why it needs the blastocele, is for these initial movements to give it space for the cells to start moving around. So you can see a side view here. You get these bottle cells. Now, they've shown experimentally that once the involuting occurs, if you remove the bottle cells, gastrulation will still continue. So the bottle cells are really only necessary to start the initial stages of gastrulation. After that, these movements will occur spontaneously from these other cells, even if they remove the bottle cells. Just side note, more really, but some scientists said, well, I wonder if these are necessary for continual movement of these cells, through that, and they showed they weren't. Okay, so dorsal blastopore lip. The first cells that enter into start becoming some of the endoderm. So some of the cells that start moving in and upwards, if you look at the fate map here, these are the endoderm. So as these cells are moving up, you can see that you have endoderm forming. The next cells that are coming in form head mesoderm, and the ones after that, chorda mesoderm. This is a transient group of tissues that comes from the mesoderm that forms a region called the notochord. Now, the notochord is a huge signaling center that does a lot of patterning, not only of the nervous system, but also muscle and bone and tendons and things of that sort. They found over time that some of these notochord cells don't just go away. They actually become part of regions of the vertebrae, actually in between the vertebrae, the intervertebral discs. But they used to think that the notochord was just there, and then the cells dissolved and went away, and they didn't contribute. They're now finding that some of those cells actually do contribute to the connective tissue between the vertebrae. And then the last thing is the yolk plug. Is as these cells are involuting underneath here, then the ectoderm starts undergoing epiboly, where it undergoes some mitosis, and the cells start flattening out. In fact, it starts out really thick and eventually gets thinner as well. And the reason why it gets thinner is because the cells are pushing out and forming a much thinner layer. It's almost like taking 5 lanes of traffic and squeezing them into 2. It's much thicker at first, but as you squeeze them into 2, it's going to get much, much longer. So that's one of the factors that are involved in here as well, is that the ectoderm initially starts out really thick. As it undergoes epiboly, it gets thinner as the cells start pushing around and enveloping the entire mesoderm and endoderm. So that's how this internalizes all of the cells that are mesoderm and endoderm, so that the surrounding tissue is just ectoderm, and then the mesoderm and the endoderm will start forming the various other parts of the organism. This just kind of shows you some actual slides of representing the same cartoon pictures you just saw here. Here you can see the dorsal blastopore lip, these cells that first come in. You first have some endoderm, and then you have the head mesoderm, and then the chorda mesoderm. You still have endoderm here. These cells will involute as the ectoderm will undergo epiboly surrounding it. And here's another illustration showing you how all of the cells will pretty much envelop and finally finish surrounding that yolk plug and then connect together, and then you'll start getting the patterning of the neural tube. So lots and lots of animations or slides here, just showing you the same things I've been going over here. In zebrafish gastrulation, one of the first movements is epiboly. So epiboly begins first of these blastoderm cells. And when we say the blastoderm cells, these enveloping layer, the enveloping layer right here is what starts to begin as far as the epiboly that will ultimately surround the yolk. And the deep cells, this is what eventually will become the embryo proper, or the actual embryo itself, whereas the enveloping layer doesn't actually become the embryo, but does take place in the gastrulation movements as well as in its specification. Now, one of the first things you have to know is this outer layer of cells, we call it the embryonic shield, just like a shield protects and coats. The embryonic shield is what undergoes epiboly. So as these cells start undergoing convergent extension, remember I explained convergent extension is kind of like taking 5 rows of cars on a freeway and squishing them down to 2. As you move them down to 2 lanes, it lengthens out. So it goes from a thicker layer to a thinner layer, and that's why these cells are able to move and ultimately envelop all of the yolk area. So you can see that as epiboly occurs over here, you'll also start getting involution of cells that will start moving underneath the sheet of the embryonic shield. There is a difference here in the gastrulation movements in terms of what becomes what in the germ layers. What I mean by that is I told you in human and in chick gastrulation, the hypoblast, which is the sheet of cells that forms underneath the epiblast, doesn't become any of the germ layers. Well, that's different in Xenopus. In the zebrafish. In the zebrafish, the hypoblast does become the mesoderm and the endoderm. So this is different than the others in that the hypoblast actually becomes those 2 germ layers, whereas in chick and in human gastrulation, the hypoblast is necessary for patterning, but it does not become any of the 3 germ layers. So that's a big difference between them. So here's a side view. Here's the embryonic shield, the enveloping layer, the deep cells that will become the embryo itself, and then epiboly will cause this to ultimately surround that. Here's the yolk syncytial layer. Eventually, these nuclei will be just under the periphery of the embryo as it's developed. So these cells won't really go much deeper into the yolk, but they will still be yolk syncytial nuclei. Let me show you how that is a little bit later on, like this. So you can see how there still are some. And these are necessary for patterning and for induction events as well. Over here, we have the dorsal region and the ventral region. Now, this is determined by maternal components and certain genes we're going to get into later on today when we talk about axis specification. So here is where you start getting the hypoblast starting to form. You can see how the hypoblast will actually form in both of these areas. However, this region right here plays a key role in patterning. We're going to talk about in just a second what we call the organizer. So just keep that in mind as we talk about this. This dorsal region right here plays a particular role. Now, the hypoblast, as I mentioned here, does become the mesoderm and the endoderm. The epiblast here, as you can see, will become the ectoderm. So the epiblast here becomes the ectoderm, and the hypoblast becomes the mesoderm and the endoderm. That is completely different than the chick and the human. So we'll show that here in a second as far as the gastrulation. So here we have involuting cells. This is the equivalent to, in the Xenopus, the dorsal blastopore lip. It does the same types of initial gastrulation movements as it does in the Xenopus, where you start getting involution of these cells. And at that same region, you also get the organizer. You'll see how in each one of these models, even though they have some fundamental differences in their modes of, in some of their patterning, some of the same processes occur in every single one of them. So, involution of cells, you have the epiblast. These involuting cells will become the hypoblast, and then convergent extension causes these outer cells to essentially extend and undergo epiboly to surround the entire yolk layer. You'll still have some yolk syncytial nuclei that will be just beneath this layer that are necessary for further patterning. So here you start to see the establishment of the various germ layers, the ectoderm on the outside, the mesoderm, and then the endoderm. Most of the hypoblasts will become mesoderm. Endoderm, it's a little more complex in how it forms, but it does form from the hypoblast, but a good majority of these initial cells that are forming the hypoblast will ultimately end up to be the mesoderm, as you can see here in this layer as it forms. So those are the initial stages of zebrafish gastrulation and setting up of the body plan. Let's talk about chick gastrulation. We talked about it forms on top of the yolk. It is meroblastic. It does form a hypoblast. So first it forms an epiblast, and then it forms what we call subgerminal cavity through osmosis, where it causes fluid to go between the epiblast and the yolk to form a barrier, just like the others. Now, that's not technically the blastocele yet. Once the hypoblast forms just beneath the epiblast, the layer in between the epiblast and the hypoblast, that's what we call the blastocele, similar to the Xenopus blastocele, as we saw previously. Subgerminal cavity, yeah. So the subgerminal cavity is just the fluid-filled area. That's what makes it so that this middle area is a little more translucent, which is why we call it the area pellucida, and the outer area is opaque because the cells don't have any fluid between that layer and the yolk. So here is a side view. You've got the epiblast forming on top of this area. Here we've got the subgerminal cavity, so you already got a lot of fluid. Here are cells that are confluent or touching the yolk area, so you don't have space in between here. The yolk is just beneath right here. But this region plays a critical role. Now, one of the methods of how the hypoblast forms here is it doesn't form necessarily from cells extending out from these cells right here, but rather from the epiblast cells will ingress. Actually, we typically call it delamination. If you remember that gastrulation movement, delamination is where one sheet will actually form into two sheets. So these cells will undergo an epithelial to mesenchyme transition. They'll break free from the epiblast, they'll migrate downwards, and they'll form a new layer. So we call it delamination because it forms a new layer or sheet of cells just beneath the other cells. Now, the space in between the epiblast and the hypoblast, that is the blastocele, and that is necessary for the gastrulation movements that are going to occur here in just a little bit. So here's a top view looking at these regions. The top here, looking at the dorsal side or downwards, you're seeing the gray, which is the epiblast. Underneath, you're looking at the hypoblast. So when you're looking underneath at the ventral side, this is just showing how the cells start forming the hypoblast, this sheet of cells just beneath the epiblast itself. Okay? Now, in this situation, the hypoblast does not form mesoderm or endoderm. The epiblast completely forms all three germ layers. The hypoblast, however, is necessary for patterning. That's why one of the biggest processes of today we're going to talk about is axis specification and differentiation of cells and whatnot. So the hypoblast plays a key role in the patterning of the three germ layers, even though the hypoblast itself does not become part of any of the germ layers of the embryo. I'll talk a little bit later on about the primitive streak and Kohler sickle, but one of the, in terms of patterning. But what happens is you're looking at the area pellucida here. In the most posterior region, cells will start to invaginate. Okay? Remember, the invagination is just where they start infolding and forming a groove. This invagination is, again, equivalent to the dorsal blastopore lip in Xenopus, as well as that embryonic shield in zebrafish. Now, you don't get involution yet, but you do get invagination. So it starts forming this pit where the cells start kind of forming a little groove. In fact, we call it the primitive streak. Sometimes we call it the primitive groove because it's a tiny invagination. And it extends from the posterior region all the way to the anterior region. So you actually see the groove start moving forward towards the anterior region of the area pellucida. And that's going to start forming the axes of the chicken. So eventually, there comes a point where you see this long groove, this pit, this invagination of cells. They don't invaginate all at once, but it actually moves forward, and the cells move forward to the anterior region, and this as well. You do get some convergent extension where you get elongation. You can see how initially the area pellucida is circular in nature. Eventually, it starts extending out, just like you saw with some of the other model organisms. If we look at a fate map, you can see that at this point, we can mostly determine what ectoderm, neural ectoderm, as well as various types of mesoderms are going to form. We don't see endoderm yet at this point, but you will see that here soon enough. So the primitive streak plays a key role in the anterior-posterior axis formation. In fact, when you look at it, this also is where left-right patterning starts to come into play. And there are various genes that get turned on the left side that aren't turned on the right side, and that's what makes the heart on one side and the liver on the other side, and so on and so forth. That's where you get that left-right symmetry. Okay. You can see further elongation of the embryo as it undergoes extension. And there are a lot of mitotic events going on here, too. We're going to get into some of these things. What I want to illustrate right here is this. Now you start to get cells that are involuting. Actually, we would call this ingressing because they do break free and become mesenchyme. But then some of the cells will reform a new epithelial layer that will form the endoderm. So you can see here in the green cells or the hypoblast, what happens is as the cells start ingressing in through the primitive streak or the primitive groove here, they will undergo this transition from epithelial to mesenchyme. Some of the cells will migrate inward to the middle area, and that will become the mesoderm. Other cells will migrate further ventrally, displacing the hypoblast. So these cells will actually push the hypoblast away and form another layer of cells beneath it, and that will become the endoderm. So that's why all three germ layers come from the epiblast. The hypoblast is necessary for initial patterning, but eventually cells from the epiblast will displace the hypoblast and start forming endoderm, and other cells will start forming mesoderm. Now, this is the same way in which humans begin their initial stages of gastrulation. We have this patterning of the epiblast and the hypoblast. The cells will undergo the primitive groove. Cells will start ingressing. They will move in and form mesenchyme in the middle here. Again, remember, this is the blastocele, which allows cells to have that ability to move and push things out of the way and migrate and gives it that space necessary. And then the hypoblast gets displaced and by these cells that are ingressing, and this will form a new sheet of cells, which will become the endoderm. Those are the initial stages of gastrulation. Eventually, you start getting the head forming and the tail forming, and this is where the brain's going to form and the spinal column, and we'll get into all that good stuff later. Let's look at human gastrulation, since it's so similar to chick gastrulation. Here, we've got the trophoblast cells that are on the outside. Here's the inner cell mass as it's been patterning during its journey down towards the uterus. Eventually, you form the epiblast and the hypoblast, identical to what you find in the chick gastrulation. The same thing is true. The hypoblast does not become any of the germ layers. It doesn't become mesoderm or endoderm, but it is also necessary for patterning. This is one of the reasons why using chick gastrulation as a model for human gastrulation teaches us so much about ourselves. If you're looking at a dorsal view, you can see the same movements going on, where the cells will ingress, and a lot of these cells will move forward to form where the head's forming. Here's a transverse section looking at it sideways. Again, here's the hypoblast beneath, and as cells ingress, they will form mesoderm and displace the hypoblast to form the endoderm. So all three germ layers, again, come from these epiblast cells. So comparison. Let's compare these models for now. They're actually very similar to one another between chickens and birds and frogs. As we talked about, the blastocele is always between typically the ectoderm and the mesoderm and the endoderm. So here in the frog, the Xenopus, you have a huge blastocele, which plays an important role in patterning. Over on the top here, you have the ectoderm, and these two layers are going to be the mesoderm and the endoderm. Over here, we find the epiblast and the hypoblast, and in between that, you have the blastocele. Over here in humans, or in mammals in this case, you have epiblast, the hypoblast, and then you have the blastocele in between. So some of the same movements occur here. In fact, we didn't show Epiboly, but Epiboly does occur where the ectoderm, what will become the ectoderm, will surround the mesoderm and the endoderm. So at a certain point in gastrulation, as these 3 germ layers are being set up, this outer layer will completely undergo Epiboly and engulf and internalize, as we mentioned before, is one of the processes that happens in all of these gastrulations, internalizes the mesoderm and the endoderm. So Epiboly is occurring, but we don't really see it at these initial stages like you do with the Xenopus or with the zebrafish. All right, organizers. Now we get into the big one. This is key to understanding how cells start forming the 3 germ layers. Because as we've mentioned up until this point, this is where the cells take on different fates. This is where they start going down different pathways, different differentiation pathways. And it's all about the paracrine factors, the juxtacrine factors, the morphogen gradients, and all of those things that they're exposed to during these initial stages of gastrulation. So in reality, organization, or the organizers, or the tissues that are patterning these cells, they're merely groups of cells that are typically releasing paracrine signaling to adjacent cells that tell them what to become. They're typically under autonomous specification, although in some cases there is reciprocal induction going on between the organizer tissue. But we'll find that in each one of these cases, some of the same paracrine factors and methods of organization are found in every single model organism. They're the same, which is why it's so good to study them, because we can learn, again, about how we are patterned. Now remember the axes that we're dealing with. Anterior, posterior is head to tail. Dorsal is the back, ventral is the belly, and then left, right. These are the 3 axes that we typically look at when dealing with the patterning. So let's talk about Xenopus first. This is one of the more highly studied organizers amongst the 4 vertebrate model organisms, although chickens have definitely been studied quite a bit too as far as their organizers. So here is the universal concept about organizers. Organizers have the ability to self-differentiate typically into dorsal mesoderm. So in many cases, they can receive their own inductive signal. They're kind of self-differentiating to be able to release, what do we call it, autocrine signaling, as we've mentioned. They have the ability to dorsalize surrounding mesoderm as well as into paraxial mesoderm. So they release various paracrine signals to cause mesoderm to become dorsal. You'll find that these organizers are typically in the dorsal region. They will also are necessary for the dorsalization of the ectoderm, and most key is their ability to initiate gastrulation. In fact, everything we've talked about up to this point, the involution, the ingression of cells, those are all initiated by the organizer in each one of these organisms. Okay? So let's look at how these things work. Now, they've shown that at a certain point, if they remove cells from one part of the tissue, do a transplantation experiment, if they take it from various regions and put it into another region, nothing happens. The cells are not committed at that point to become ectoderm or to become neural tissue or whatnot. However, they find that at a later stage, if they take the same tissue that's been affected by the organizer and move it to another area, it starts forming neural tissue where it shouldn't. So there comes a point where once the tissues or the cells have been influenced by the organizer, they start to become more committed down a particular cell fate, whether they're going to become mesoderm or endoderm or neural tissue, which is what they show here. The same tissue, before it's influenced by the organizer, hasn't committed itself to what it's going to become. So you move it to another area, it's going to become a different tissue. However, in this point, these cells have been exposed and undergone paracrine signaling, You move it to another part, and now it becomes a second neural plate and forms another neural tube. In fact, an extreme case of this, if they take the actual organizer area itself and transplant it to another area, guess what happens? You form two whole embryos. That's how powerful the organizer is it has the ability to pattern the three germ layers even if you move it to another area, and you end up getting conjoined Xenopus, or conjoined frogs, essentially, in this case. You get different neurulation events, and it essentially patterns the other part of the embryo as a result. That's how powerful the paracrine signaling from the organizer is. If you were to take this tissue and transplant it to another area, it would organize a whole other embryo as a result. So let's look at the fate map of a Xenopus and look at the organizer's influence over that. One of the important things that we've talked about here with the Xenopus is the blastocele. And the reason why this needs to be here is because the organizer, which is typically found right here at the dorsal blastopore lip, as it secretes paracrine signals, if this is confluent or touching the yolk, if there's no blastocele, no ectoderm forms. It actually forms into mesoderm. So whereas these cells would normally take on an ectoderm fate, if you were to remove the blastocele and just put that tissue right adjacent to it, it would form mesoderm. You would get no ectoderm. That's why the blastocele is necessary, to separate it so that it's not influenced by some of these paracrine signals that come from the organizer. So let's look at how the organizer forms. There's actually two different areas that we have to talk about in relation to that. There are maternal factors that come into play here that are necessary in the formation of the organizer. In the animal pole, in the vegetal pole of the Xenopus, there is typically a sequestering of a protein called VEG1. And this protein, in combination with another protein, forms a group of cells which are called the nucleus center. Now, this is not the organizer. However, they showed that the nucleus center induces the organizer. So these cells release paracrine signals that will then tell these cells to take on that fate of then releasing other paracrine signals. So the organizer doesn't form until the nucleus center sends out these paracrine signals to cause it to form. And they've shown this through a number of various transplantation experiments on how you get this induction process, which I'm not going to go through for this. However, one of the things I want to show you as far as axis formation is you get morphogen gradients. Here in the ventral region of the Xenopus, you get high levels of bone morphogenic protein, or BMPs, and then you get low levels towards where the dorsal end is at. Here in the posterior end of the Xenopus, you have Wnt signaling. You have high levels of this morphogen factor, and then it gets lower and lower as it gets dispersed through the vegetal pole. So here, the maternal components, you have BMPs on the ventral side, and you have Wnt on the posterior side, and that's how the anterior-posterior axis typically is determined, and the ventral-dorsal axis is typically determined. Now, in order to establish those gradients, we have to first talk about fertilization. I told you this was going to come back into play. One of the things about Xenopus is, as it's set up, in the most vegetal region of the vegetal hemisphere, disheveled gets sequestered. You have disheveled protein. Now, you actually have the protein. You don't necessarily have the mRNA. You have disheveled protein here that's bound. When the sperm fertilizes the animal pole, then you get the cortical rotation. So what ends up happening is that cortical rotation pushes the disheveled to what will eventually become the dorsal region of the embryo. Now, as that protein then gets released, it forms a morphogen gradient where you have high levels of disheveled in half of the embryo and no disheveled in the other half of the embryo. Well, guess what that does? If you remember the Wnt signaling pathway, disheveled blocks a protein called JSK in this pathway. JSK is or JSK3 is the protein that degrades beta-catenin. So in the area where disheveled is found, you get beta-catenin surviving. You don't get degradation of beta-catenin. Over here, on the other side, where there is no disheveled, beta-catenin is degraded. And that then changes. This kind of creates two hemispheres, and where half of the embryo has beta-catenin stabilized, the other half of the embryo has no beta-catenin in it. Well, guess what? You've already established that in the vegetable pole you have other morphogen factors, such as, let me show it right here, VEG-T and VEG-1. When those cross over with beta-catenin, this becomes the new coop center. So here, where beta-catenin and VEG-T and VEG-1 cross, that's the new coop center. So guess what happens there? Certain genes are turned on, and then paracrine factors are released, and that induces the organizer. That's how the initial body plan gets set up in the Xenopus, is the cortical rotation causes beta-catenin to be moved over to this side, the dorsal side, and then you get this kind of crossover between beta-catenin and VEG-T and VEG-1. That then turns on key genes. If you look back here, turns on SMAD, signal transduction pathway, which then turns on other proteins like Discoid. I mean, I don't want you to go through all of the various genes that it turns on. There's quite a few, and it's very complex. But the fact of the matter is, when you get a combination of beta-catenin, which is necessary for the transcription of key genes, and the combination of VEG-T and VEG-1, then you turn on key genes in the new coop center, which then initiate transcription of key genes in the organizer. And that's how the organizer gets formed. Another set of genes that get turned on in this combination is called brachyury, or XBRA, and these will induce this region right here to become mesoderm, most of the region to become mesoderm. And so there's a lot of patterning going on here. There's Dickoff, as I told you. We'll talk about Dickoff later on, but that's how it's spelled. It's a German word. Okay, so that's how fertilization actually starts influencing the initial stages of the Xenopus setting up its body plan. So let's talk about what the organizer does. The organizer releases a number of proteins. You can see how many different proteins are happening here. Well, in where the organizer is at, you have blocking of BMPs. So over here, you get this grading of BMPs, but where the organizer is at, it actually blocks bone morphogenic proteins. That's one of the things that it does, is it prevents BMPs from being expressed in these other regions right here. So here, the organizer is going to block the BMPs. BMPs are going to be found all throughout the ventral region here. In fact, the default of the ectoderm is to actually become neural tissue, but when you get BMP signaling, it actually causes the ectoderm to become epithelial, or not epithelial, it becomes the skin and surrounding tissue, so the epidermal layer of the skin and that outer region. So in the area right next to the organizer right here, this ectoderm is actually going to become neural tissue because BMPs are blocked by the organizer. This is a key component of what the organizer is doing. Another thing that it does is it causes the head regions to start being able to be formed. So I'm not going to get into all the details of all of the proteins that the organizer forms. But I want you to know this is also where the dorsal blastopore lip forms. So gastrulation, or the initial movements of the cells, are initiated by this organizer region. It causes the bottle cells to come in, it starts the involution of the cells, and it initiates the epiboly that will start surrounding and engulfing the mesoderm and the endoderm from the embryo. So here is just a look at some of the signaling that's going on here. Here, the organizer, one of the proteins that it expresses is Dickoff. If Dickoff is blocked, then the head won't form. But if it is expressed, then you get the head region forming, and you get the neural tissue forming, and so on and so forth. Let's look a little bit about just the two major players in differentiation here. This is where we get into cell specification. The ectoderm, as you should know, becomes not only the outer covering, but also becomes neural tissue. Well, depending upon what the ectoderm is exposed to, there are several fates. For example, if the ectoderm receives both BMP and Wnt signaling, it'll become the epidermis. It'll become the skin. It'll become the outer layer. It'll become, feathers for chickens and other things like that. Here, if BMP is blocked, as it typically is done by the organizer, but Wnt signaling is present, that becomes the spinal column. And in the region closest to the organizer where you have both BMP and Wnt signaling blocked, that forms into the head ectoderm. And there are various key genes that the organizer secretes, like Cerberus and Dickoff, that block BMP and Wnt signaling. So this is just one example of many different ways in which paracrine signaling will block certain signal transduction pathways. Over here, these are allowed. Near the organizer, they're not allowed. BMP and Wnt signaling is not allowed, and that allows this ectoderm to become neural tissue specifically for the head. Now, one of the things they found in vertebrates that also plays a key role in anterior-posterior patterning is retinoic acid. This is going to come up quite a bit. Retinoic acid is a key factor in turning on many of the Hox genes. And so the Hox genes definitely play a key role in anterior to posterior patterning. So just keep that in mind, that retinoic acid, the gradient, it actually has a low to a high gradient, and that forms the anterior to posterior axis. Let's talk about fish. In fact, this is actually a little bit easier. In the initial stages of the fish or the cleavage of the zebrafish, there is a difference in where the maternal components are. In fact, in this area where gastrulation begins, there's a key gene. Let me jump ahead to it right here. It's called nodal. So nodal is sequestered to this region. If you're looking at the cleavage of the zebrafish here, you can see in this region you've got some sequestering of nodal, where you have a high amount of this protein. This is where the organizer is going to form in the zebrafish. This is also where gastrulation begins, in that embryonic shield, where you start getting involution of these cells. Remember, the organizer is responsible for initiating gastrulation. So here in the dorsal region, over here is the ventral region. This is where the organizer is going to form. One of the key proteins that are responsible for that is NDR1. Okay, let's come back here. So here, right here, this embryonic shield, this is the dorsal region. This is where the organizer forms due to nodal, and this is where involution of the cells begin. We already went through the gastrulation movements here. So let's look at some of the other factors going on here. The same thing occurs. You get combination of beta-catenin and nodal in this dorsal region, and that's going to also create the organizer in that region of the zebrafish. So the same proteins, beta-catenin, nodal is similar to the VEG-T and VEG-1, essentially. So let's look at how this works. That's the anterior-posterior pattern. Here we go, dorsal-ventral. So looking at the top, here's the ventral, here's the dorsal. You can see that over here in the dorsal region is where the nodal is at. This is where involution of these cells is going to begin. You get Epiboly of these tissues going around, but this is what's going to start gastrulation and involution. Also, as the hypoblast is forming, they're going to be exposed to various paracrine factors by the organizer, and that's what's going to cause the hypoblast to start forming the mesoderm components and eventually the endoderm components. So the signals received, the paracrine signaling received by these cells as they move through and start involuting is what's going to help them take on their particular fate. Cells or genes are going to be turned on as a result of these paracrine signals. In fact, the paracrine signals change over time as cells that come in late will be exposed to different signals, and they'll actually take on a different fate. Again, retinoic acid plays a key role in anterior-posterior patterning of the zebrafish. You don't necessarily have to know all the dynamics behind this, but retinoic acid again, is another factor in anterior-posterior patterning. It increases as it goes from anterior to posterior and turns on key genes for that patterning. Left-right. We didn't talk about left-right patterning in Xenopus, but one of the things that happens in left-right patterning in zebrafish is there are these ciliated cells. And due to the ciliation, the cells will actually sweep in a counterclockwise fashion, and it pushes certain molecules over to one side, and that's where you start get the initial formation of left-right patterning. And so this creates a distribution of different genes on one side or another, and that's one of the things they found that will start the left-right patterning that's necessary, is that these ciliated cells will actually rotate in a counterclockwise fashion and push molecules to one side that will then begin that patterning. Pretty fascinating. So these are the three axes. Anterior-posterior patterning is primarily by FGF Wnt signaling with retinoic acid, similar to what you saw in the Xenopus. High amount of Wnt signaling is in the posterior end. Low amount of Wnt signaling is in the anterior end. Retinoic acid plays a key role. As it increases, you get more posterior genes being turned on, and that causes the tail end to start forming, whereas little retinoic acid is in the anterior region, and that forms the head. All right, let's talk about chicken embryos. This one's actually easier than all the others. So, the organizer in the chickens is called Henson's node, after the scientist who actually discovered it. His last name is Henson. So, Henson's node. This is the equivalent of the dorsal embryonic shield for the zebrafish, as well as the dorsal blastopore lip in the frogs. You can see how they all play the same role. They're all for the initial gastrulation movements and the necessary paracrine signaling to start setting up the body plan. So when does Henson's node form? Well, let's look at the chicken embryo. Here is the comparison between the frog, which we just went over, and the chicken. In the orange dots is beta-catenin. So we know how cortical rotation causes the embryo to shift, and then you get beta-catenin in one half of the frog embryo and no beta-catenin on the other. Here's the vegetal pole of the frog embryo, where you get veg-T and veg-1, and then when you get the crossover, that's where you get the nucleus center. Well, let's look at the chicken embryo. Instead of having beta-catenin to one side or the other, beta-catenin is just around the periphery of these cells, of these epiblast cells that are forming. Here, in the most posterior end, you get crossover between beta-catenin and veg-T and veg-1. That forms the nucleus center. So same thing here, just this is where the posterior end of the embryo is. Remember we talked about how the primitive groove starts forming from the posterior end? It's right there. It's at this junction, and that's why these cells initiate the gastrulation movements, is because of the beta-catenin and the veg-T that initiate the gene transcription that's necessary for the changes to occur in these embryos. Now, how does the veg-T get sequestered over here? In fact, that is primarily due to gravity. As the egg is going through the oviduct of the chicken, as it's actually rotating. And due to that rotation, the gravitation, it pushes the veg-T to the most posterior end of where these cells are undergoing cleavage. And so that's how veg-T actually gets pushed to this area, and that's what creates the most posterior region where the organizer is eventually going to form from this nucleus center. So gravity plays a role in the posteriorization and the anteriorization, so anterior-posterior axis of the chicken. So in the actual laying of the egg is part of the anterior-posterior patterning of that. If you just pulled the egg straight out of the chicken, it wouldn't work, well, for a number of reasons. So let's look here. This region, the nucleus center, will induce the various cells that will eventually become Henson's node, which is the organizer of the chicken. Now, Henson's node doesn't actually form, the organizer doesn't form until these cells reach the most anterior region of the embryo. So as the primitive streak's forming, these cells are being exposed to genes from the nucleus center, and eventually these cells will take on the organizer fate. Okay? So Henson's node doesn't show up until the entire primitive streak has been developed, and then in the most anterior region of here, you get Henson's node. That's where the organizer forms, is in the most anterior region. Here we have various genes. Now here you're looking on the green region right here. This is the hypoblast. Remember I told you that the hypoblast does not play any role in the germ layers. However, it does play a key role in the patterning process. So here we see we have FGF signaling and Cerberus and other signals that are inducing the epiblast as it starts undergoing its gastrulation process.